I think we're open and just about ready to go here. I think we have quite a good crowd of people here now joined. So welcome to this session on sharing of our data on COVID research and photon neutron facilities. I hope this will be an interesting session. So I'm Brian Matthews, so I'm going to do some sharing today. So I thought I'll just give a couple of words on what we're looking at here in this meeting. What we're looking at is data sharing, COVID research, and photon neutron research infrastructures. Photon neutron facilities have played a very significant role in COVID research over the last year. All the facilities that I know of have sort of realigned themselves quite rapidly over the last year to emphasize COVID research and prioritize it. So photon neutron facilities are a real engine for doing COVID research and have generated large amounts of data, and then there's an imperative to make that accessible, interpretable, and interoperable with other researchers so we can share it more easily, so we can speed the science. And this should fit in with wider efforts to make data fair for facilities. In an RDA context, there was the RDA COVID-19 working groups got set up last year to look at making joint recommendations on data sharing, and this is a link to their report that came out last year with lots and lots of very extensive report, lots of recommendations. Some of the recommendations directly addressing the research that involves photon neutron facilities, and others sort of part of the wider environment in which our research sits. So this session, I felt, was an opportunity to reflect on the operation, on those recommendations, share approaches to photon neutron COVID experimental data, and consider if there's any further steps we might want to take to make common recommendations via the RDA or possibly elsewhere in projects. And I should mention actually projects. This session is part sponsored by two European projects, Expands and Panosk, who have helped provide us support for doing this event. This is the agenda. Got quite a packed agenda with a number of short talks of sort of 5 to 10 minutes each, and hopefully have some time for some questions in the end. Please put questions in the chat box or in the chat box and a Q&A box. I've seen people put questions in each, so we'll try and monitor both, and we can hopefully maybe have time for the odd question as we go through, but certainly hope to have some time for questions and discussion at the end. So with that, I'll stop sharing and ask our first speaker, who's Abigail McBurney from STFC, to give a presentation on fair data and facilities. And as we're not speakers, could we turn off microphones and videos if you're not speaking?
Thank you, Brian. So can you all see my screen there? Brian?
Yes.
Yes, you can.
If you can make the screen full screen by clicking on the icon on the bottom right-hand corner. You can make the image
for people who are watching. Yeah, that's great.
Watching,
yeah, because I take up half the screen. I wish I didn't, but there we go.
Yes.
Okay, thank you, Brian. So I'm going to go ahead. I'm Abigail McBurney, and I work at STFC and also in Expands. And I'm going to speak today about fair data in photon and neutron research infrastructures, or PAN-RIs. So as Brian mentioned, there are 2 Horizon 2020 funded projects in the moment in Europe that are looking at fair data for PAN-RIs. We are aiming to develop, enable fair PAN data services and to get these into the EOSC, which is the European Open Science Cloud. The similarities between the 2 projects are that we're looking at FAIR, we're obviously focusing on EOSC, and we do support the same community. But there are some differences. So Expands, if you can see the diagram on the left-hand bottom side, we focus on national PAN-RIs, whereas PANOSC focuses on the pan-European ones. The tasks and description of work are different in the two projects, and the timing is different. So PANOSC was first. So we do work together as projects, but not necessarily in parallel. So what does FAIR look like in the Pan-RI context? What are the components, and as implementation, what does that look like? So in the pink arrow on the bottom, you can see across all of this, these 5 areas, we've got FAIR, EOSC, and sustainability. They really need to be a part of all of these 5 areas that are involved in FAIR at PAN. So we have, first of all, the framework of policies, practices, and guidelines to enable FAIR. These are things like data policies, metadata frameworks, what persistent identifiers are we going to use and how are we going to connect these, data management planning, especially active DMPs, and how can these benefit PAN-RIs, and FAIR assessment and certification. So how will we know our data are fair, and how can we show others that they're fair? In terms of data catalog services, we're aiming to federate these up through the EOS so that researchers can access this through one single point. So to that end, we have developed a PAN search API, and we're working on ontologies as well. And we're also making use, I think, of non-PAN specific, but very well-established tools and elements of the wider research data management ecosystem. So for example, OAI PMH, and getting things into B2Find or OpenAir, and also looking at data, thematic data services. Data analysis as a service is increasingly important due to the volume and the complexity of the data that we're generating at PAN-RIs. But the process of analysis is a very good opportunity for rich provenance and context information that we need to help make our data fair. And it also brings in a lot of related research objects. So things like software and notebooks, these feature very heavily. Training, in our 2 projects at least, we are producing a PAN training platform, again, which will be accessible through the EOSC. And this will include not just technical training, but also training around FAIR and research data management. And of course, there's a lot of advocacy to do. FAIR is not well known in the PAN community yet, so we need to raise awareness of it both within our own facilities, but also within the user community. We also need to make sure that we fit with EOSC, but that we're also helping to shape and develop it by feeding into it. And of course, around this, you have the more general things that are needed to support this in the longer term. So the management and appropriate resourcing, but really also a strategy for FAIR. And that's what I'm going to talk about in the next few slides that I have left. And that this strategy needs to be based on the reality of where we are now to some extent. And in work package 2 of Expands, where I work in, we have this concept of data that is fair when it leaves the facility. Because at the moment, without a really strong FAIR research culture, we can really only control what happens to data in terms of fairness when it's using facility resources or our systems. When the researcher takes it away, we have less control of what they do. So I have some questions around particularly our strategic approach to FAIR and PAN, and I call these are my thoughts, and others may disagree, but I call these my elephant in the room questions. There are probably more than 3, but I'm focusing on 3 here. So unlike many of my Expands colleagues, I don't come from an IT or a PAN science background. I'm an analyst and an information specialist. So to me, these are really important questions, these 3, especially from the long-term strategic perspective in relation to FAIR. And it's really not true that we're not addressing these questions, but they're challenging questions, difficult questions. And it's really just all too easy to turn away from these to focus on something more IT related, for example, or something that's a little bit more urgent to do in our facilities. So the first one is around the fact that we have the same user base, but PAN-RIs differ. And how do we deal with this diversity? So this may not seem very relevant to FAIR in the beginning when you first look at this, but it's important to remember that there is no one way of doing FAIR. So we should remember this and value, I think, the diversity and the differences in approach that we have, and aim to accommodate and learn from these differences across our PAN-RIs. Obviously, we don't want to confuse our users. It needs to be moderated in that regard, but we have a lot of variety in approach, from legacy, from natural research culture issues, all kinds of things. And I think instead of seeing that as potentially as a problem, we should see that as an opportunity. The second point here is that FAIR is about being selective. So as a research data management position in PAN, I think our default position has basically been, we have the space, so we'll store it. That's kind of been our RDM approach. And the focus is really very much on storage and access historically. And this makes sense because we're trying to make sure our users can access their data. But FAIR is really more about data curation. And data curation is about the value and usefulness of data throughout its life cycle, and also considering the end of its life cycle. And part of this is being selective. And so I think when we think about FAIR in our facilities, we need to switch from this storage and access RDM model more and more towards this data curation idea. And that will help us make decisions that we need to make around FAIR. And finally, metadata. So metadata, information about data and other things, is really important when it comes to sharing. But what is our ambition in this regard? Not just our aim, but our ambition. What do we really desire and want to accomplish with this? And I'm going to talk about this in my next 2 slides. So the first point to make is that we have a lot of metadata in pan science. This is a model that a colleague in Expands has made. And if you look at the middle ring there, you can see a whole range of metadata. And this isn't necessarily even everything, but it gives some sense of the kind of richness that's available. And one of the questions I think we need to ask ourselves is, at the moment, what of this model are we actually benefiting from? What are we capturing? What are we using of what's actually available? But when it comes to fair data sharing, the idea of different levels of metadata really matters. And the reason for this is that the metadata required in any given situation depends on the users and their intentions. So in Expands, we think about this in 3 levels generally. The first level is this general browsing and discovery, and it doesn't even need to be by a researcher. This is really your basic descriptive information, Dublin Core, data site, that type of thing. And with our historical focus on storage and access for our own users, we're really most comfortable, I would say, with this level in PAN at the moment. And obviously it relates very well to the F and the A in FAIR. Level 2, I'm going to come back to in a moment and move on just now to level 3. So level 3 is around allowing a PAN researcher not just to understand a PAN data set, but also to ask questions and get answers from the metadata associated with that data set. And research has consistently shown that the most common form of research data reuse is this within the actual research team that generated the data. And because pan science is usually a team effort, this really matters for us. And here at this level, it's really about the richness of the information. So in pan, we have Nexus, for example, to capture quite a lot of rich information. And I would say in pan that certainly in the projects Expands and Panos, this is the kind of area where I think a lot of our metadata potential and the work that we're going to do is likely to be at this level for the moment. But going back to level 2, I think this is a really, really important level for us. And this is around interoperability. And we tend to think of this as, systems or file formats or things like that. But I'm going to talk about human semantic interoperability. So here we have the non-PAN researcher who wants to understand if and how they can use a PAN data set. They have a solid research understanding, but basically they don't speak the language of PAN. So I've often heard it said in PAN by researchers, oh, well, no one's going to use my data because they won't understand it anyway. And I just don't think this is good enough. And one of the reasons for that is, as my work as an analyst, I've looked at PAN research outputs. And I can tell you, for a start, that PAN-RI is published in hundreds of research areas. It's not tens, it's hundreds. So there is already a multiple of languages being spoken in PAN-RIs. The question is, are we speaking them well enough? The other point is that I've done analyses on multiple occasions for principal beamline scientists where I found that maybe 30, 50% of the interest in their work, the citations, other types of interest, are coming from research fields that beamline does not work in. This is an excellent example of a non-PAN researcher knocking on the door saying, please, I'd like to come in. And I think we need to make it our ambition to try and aim to let these researchers in. It's a very challenging level, I think, for us in PAN, but I think for our ambitions long-term, this is where we should be looking. So I like to think of the I as in, let them in, let people in, and the R we can think of, as I said, as about richness as well as reuse. So on that note, I'm going to say thank you and hand over to the next speaker.
Okay, thank you, Abigail. That was Very good, and really sets a context for the rest of the talk. I think in the interest of time, we will hold any questions to the end, but please put them into the chat if you do have any questions. And I will now ask Rob Hooft to join us and talk about the work of the RDA COVID-19 group and set a little context of what's happened there.
Yes, thank you. Let me select my presentation there. So I hope to see the information that this is actually showing the presentation. But my name is Rob Hooft. I work for the Dutch Tech Center for Life Sciences, the Elixir node in the Netherlands, and for Health RI in the Netherlands. But here I am representing the COVID-19 working group of RDA, which last year did a heroic effort. I put some slides together, and some of them that you see have been used actually yesterday by Natalie Harrower, so I stole them shamelessly from her. So yeah, that's too fast. Yeah. So the group last year, just after the planned Melbourne conference of RDA was pulled together by a request from the European Commission to get some ideas about how data should be shared for COVID-19 and whether RDA could give an idea. It was an extremely fast track process that was started, and normally a working group takes 18 months, as you know, in RDA. This delivered its first draft recommendation after a few weeks of operation, and it delivered the final version after 3 months. So, and this was including a complete review, internal and external review of the document. This required weekly webinars and meetings at all different time zones. So a lot of people worked throughout their weekend and throughout their nights to be able to do this. And the document was created by in total 160 active participants out of 600 people, members of the group. There was a single editorial group to make sure that the document that was created was a single readable document, and they did a lot of heroic effort to make sure that everybody condensed their output, and in total it became 143 pages, which is slightly longer than it was intended, but there was really no way of making it shorter. This was the set of working group co-chairs, but there were actually 8 groups that were subgroups of this group, each having their own set of co-chairs and guided by one of the co-chairs of the entire group to do it. So the group in its document created information about what the information problems were. So of course everybody wanted to have the data for COVID-19 as quickly as possible. However, there was then no time to make sure that the quality was up to snuff, and also making appointments about which standards were going to be used was short of time. So this is the overview of the 8 groups that were made. 4 of those groups given at the top are discipline specific, so it's a clinical, an omics group, epidemiology group, and social sciences group, each with their own type of data. And these groups there were 4 cross-section groups on community participation, on the use of research software, on indigenous data, and on legal and ethical considerations for the whole system. So this is just an overview of these 8 groups that work together to get the recommendation of. Each group actually looked at 2 different kinds of work. The whole thing is called recommendations and guidelines, and the idea was to have recommendations at the policy level and guidelines at the practical level for researchers. So at the policy level, of course, that means publishers, institutions, that means funding agencies. So there are recommendations for each of those, and there are guidelines mostly for researchers and also something for infrastructure providers. And this split is made for all of the chapters in the document. So the document itself consists of an executive summary, an introduction, some foundational elements, really the basis of the document, the extract, something that is in common between each of the chapters. Then the 8 chapters, and it also contained a glossary and an extensive bibliography. So there are some key recommendations here. I'm not going to read all of them, but if I give a biased view, then they represent a need for fair data sharing. Make as few hurdles as possible for reuse. Try to get rid of embargoes where possible. Harmonizing data standards, balancing timeliness with precision. Use data management plans. Use trustworthy repositories and document your data. And of course, all of the stakeholders that I just mentioned, journals, funders, researchers, and infrastructure providers are playing a role in this. So I think the most applicable to the PAN community are the omics group, and I happen to be one of the co-chairs of the omics group. And so we have policy recommendations, of course, for the different people, and we have guidelines, and the guidelines were split up in 5 different kinds of data. So for virus genomics data, for host genomics data, for the structural data, for metabolomics, and separately for lipidomics. I'm forgetting here proteomics. That's also was also one of the subchapters. And for each of those, we had a list of repositories, 2 lists of repositories. One primarily aimed for depositing data, so for researchers that are creating data, and the other one for a list of repositories where people can search for data. And there are some fields where this is different. Then for each field also we listed the metadata standards and the data standards. So for the structural data, this is the list of some of the subsections again. So for the data and metadata standards, we made a distinction between standards for X-ray diffraction, for electron microscopy, NMR, neutron scattering, for molecular dynamic simulations, and for computer-aided drug design data. So let me quickly try to give you some idea of what the other 7 groups did. So the clinical data, they really focused on standardizing terminology. The balancing of timeliness with privacy was one of their important issue. And also they were calling for supporting infrastructure, namely repositories. In the epidemiology group, the key was to need a harmonized data specification, including the identification of risk factors. The social sciences really called for making sure that there is enough cross-cultural data collection and collaboration. In the software group, the basic message is don't forget software, because it is essential to document your software if you want to document your data. And the legal group focused really on stressing the differences between jurisdictions and pulled towards completely open sharing where possible to minimize the hurdles that are caused by these differences in jurisdictions. The indigenous data group stressed the care principles, so the collective benefit, authority control, responsibility, and ethics principles that have been defined quite recently, and also stressed that this is important for all kinds of data that are there. So for every other chapter, this is relevant. And the community participation group called for ensuring broad participation from the different groups, including care professionals, data stewards, and the public in the collection. So next to the actual document, the 143 pages, there were 3 other ways in which the data is from the recommendation is represented. We've made a navigation tool where you can answer a few questions and get the guidance from the specific chapters that are relevant for you as a stakeholder, a specific stakeholder. There is an infographic from which I have been pulling some information in these slides, and the link to the complete infographic is here. And there is a mind map that also represents the whole piece of information, but you can navigate by going through a mind map and find the information that is relevant for you. Next to this, basically the same textual information represented in different ways, there are also many additional outputs, including one from the epidemiology group. There's a growing number of papers, including papers, a social science paper, describing the process that was used to get to this result so quickly, and also including a recent correspondence in Nature as a response to an earlier correspondence, basically asking why are there no recommendations for data sharing for COVID-19? Of course, this needed to be answered by the RDA group saying, well, there is actually. There is a very extensive and curated Zotero group library for the group that represents, I think, over 1,000 entries with information about that is located in the recommendations, the background information. And more recently, there was also a new web page created that is called The Value of RDA for COVID-19. And that's where I would like to end it, and I will hand back to the moderator.
Hello. Yes. Thank you. Thank you.
Yeah, Brian, thank you.
Yes, really good. Really useful. Do you mind if I ask you just one or two quick questions before we move on?
Of course.
Firstly, do you have any idea of the kind of impact or uptake of the work to date?
So I think the website that I mentioned, the recent website is focused on this kind of impact. I have not done of the measurements myself, but this is definitely something that is interesting for RDA indeed. Right.
And secondly, are there any plans to sort of revisit it, keep it up to date, revise it anyway as new information comes in?
So, of course, a lot of this information is not specific to COVID-19 and is actually interesting for any kind of emergency situation, most specifically any kind of infectious disease pandemic. There is an effort by people that also took the initiative for this working group to set up a community of practice for infectious disease in RDA. I can very well imagine that making sure that this document evolves over time would be a very good thing to do in that community of practice.
Yeah. Right. Thank you. And if anyone else has questions, please put them in the chat. We will get to them later if you want to say now. So thank you. Thank you, Rob. In that case, we'll now move on to a set of short presentations on giving examples of data sharing of COVID research data that's already taken place in, well, I say photon neutron facilities, all happen to be synchrotron facilities. So the first up is Lynn Pouchard from Brookhaven National Laboratory.
Yes, I'm going to try to share my slides. I had problem for it earlier, so I send them to you, Brian, and in case if I manage, maybe you can share them and I can talk to them.
Yeah, so, okay. I will check, but hopefully you can drive them.
Let's see, application window.
It's the
no, it's like a Microsoft PowerPoint. It shows blank. Let's see.
So it should say share desktop.
Yes, but I can see that. I see entire screen, and it does not show PowerPoint. PowerPoint is not recognized.
Oh.
So that's the same as it happened earlier.
Maybe I will try. I'm not seen this for on this system, so I hope this works. I will try and share my screen, and and then hopefully you can then speak to it. So let's give that a go. Does that work?
Yeah, that I see it very well. I don't know.
Okay. Well,
there's something.
Let me know when you need to move the slides forward, and I'll move them forward. Okay.
Sure.
Thanks.
Thank you. So good morning, everybody. I'm Lynn Pusha from the Computational Science Initiative at Brookhaven National Lab, which is a DOE lab located on Long Island. And this effort is, also part of my postdoc, Gilshan Park, but also many other people. And we will talk about the scientific literature sharing and analysis for COVID studies during the COVID effort. So next, please. Searching the literature for COVID, researchers have been overwhelmed by an avalanche of publication. So from January 1st to November 2nd, 2020, there was a median of 1,700 articles published per week in PubMed Central, over 57,000 articles. Compared to about 3,000 on H1N1 in the first 43 weeks corresponding of that other disease. And this is a diagram that you see at the bottom from an article in PubMed Central, and hopefully you can see it's about publishing the scientific literature on COVID. Having researchers around the world were looking for these new drugs, new treatment for infected patients, and a way to predict how the pandemic will develop. And however, nobody can read so many papers up to those several thousand articles, new articles per week. So BNL developed a literature service and question answering system using natural language processing that analyzes the papers and answers such simple questions as like, what has been written about X? Has someone reported system Y? Has anybody looked at a drug compound, for instance? Next, please. We had developed previously a scientific literature mining system for materials discovery at the synchrotron and for X-ray spectroscopy, and we were able to leverage that system for COVID. If you can do next, please. So the text and data mining system for users that we were able to leverage included automatically extracting information from scientific papers with a quick reference lookup during an experiment. During an experiment at the synchrotron, users need an efficient search system for X-ray spectra while conducting the experiment, and they may spend something like up to 48 hours at the facility. They are very well prepared, obviously, but there's always something that happens in their experiment or their sample that they did not expect, and they want to look up quickly that information and see if there's anything relevant for them in their literature. So we have come up with a domain-specific article collection that they can search. We also have a classification of papers by methods such as Zane and AXAF, by transition metal and edges. And what our system does, it extracts the figures and the caption, but also, as we realize, there's a lot of information about each figure in the text of the article, and we were able to extract sentences related to the figure in the text that are not in the caption to help users during their experiment. And we have an interface to collect the user feedback for labeled data. Next, please. And next slide. Yes, so this is a system architecture, and it's a pretty standard architecture for natural language processing. You have a list of publishers on the left, and these are slightly different for COVID. There's a lot of text pre-processing that adds metadata, remove, for instance, reviews that are not scientific articles, or and the text pre-processing is based on keywords, and sometimes the keywords only appear in references, so we remove those. And then we have two levels of the system. One powered by Elasticsearch, where the user can search directly into the articles and get highlighted sentences that correspond to their query. Or in the second system, the second venue of our website is the classification system of the papers. And then the paper can appear, the figure can appear, and then there's also those sentences that are in the paper that are not related to that are related to the figure but are not obvious from that cannot be seen. So next, please. So this is what it looks like to our user. We have the SAS classification and figure extraction. So first, the user can browse these papers, which obviously cannot be done in Google because there are way too many, and they're first classified by the methods, so EXAF, so ZANES, and then we have 30 transition metal, and for each transition metal there's a list of papers corresponding to the edges, and we get the figure. Next, please. Yes, and then for each figure we can also extract the text in the body of the paper. Next, please. And this is what it looks like. And in this figure explanatory text, we propose this to the user, and there are sentences that are directly relevant to the figure. And we removed, of course, the ones that are obvious that contain the word figure. And this is used for us to evaluate our system, where the user can actually see sentences, and there are sentences that are totally irrelevant that are being introduced in this on purpose in order that helps the evaluation algorithms to determine what is relevant and what is not relevant. And that also helps us collect better labeled data to improve the NLP algorithms. Next, please. So we were able to leverage this system for the COVID literature, and the same architecture has been used with keywords for article scrapings, and you have the list of keywords there, articles collected automatically on a weekly basis, and a list of our publishers down there. So unfortunately, these collection of articles on our systems are not available to the general public because of the licensing agreements with the publishers. So the first system on scientific literature mining is only available to BNL users, that is so people who have credentials in the BNL systems, but that includes the outside users who come to the facility at NSLS2 at the National Light Source, the synchrotron number 2, in order to access these articles. So for the COVID literature, there was a discussion with the publishers, and they agreed to make all these articles available to all the labs themselves. So not just the Brookhaven National Lab, but also agreement with all the other labs as well. And we have had 70,000 articles. Next, please. So the COVID question answering system has several components. So first, the corpus creation that collects the scientific publications, document retrieval that calculates similarity between a user question and sentences in the document to determine a relevant score, which is what we had already done for our other previous system in materials discovery, and an answer extraction to find answer to a question at the sentence level from selected papers. Next, please. This is the overall architecture of our network, and you will see on the left these BERT models. So BERT is a collection of algorithms. They're bidirectional attention models, meaning they can train the models is trained on the words that happen before a particular word or a phrase and after that word. And there's been developed a number of other subsets of BERT. That's the wrong slide. Thank you. Nope. Previous, please. Yes. So we have CyBERT, BioBERT, Clinical BERT. So these are exist as a pre-trained model, and that's a big help. So then we have our collection, and with these CyBERT, BioBERT, and Clinical BERT, and we produce a domain-tuned model. Then there's another data set from Stanford University on natural language inference that brings us to a sentence BERT. And now we have our final model properly tuned with these external resources and our data set, and we can make it active on our own COVID-19 article data set. And that allows to produce sentence embeddings for our articles that the people at the bottom left, the users and annotators, can query with their questions and give us feedbacks on the relevance of the answers that produce labeling. Next, please. That one. Yes, no, next. Yeah, this one. So the information extraction that was related to a drug discovery that we were able to extract with the system to support the drug discovery efforts. One of the crucial elements is in SARS-CoV-2 drug discovery about viral protein. And these are examples of some of the questions that we were able to ask to train our system. And the data has 114 question answers pairs provided by Ada Sedova of Oak Ridge National Lab. So for instance, what are the oligomeric states of non-structural coronavirus proteins? Are there any antivirals that target structural viral proteins? And then we have 3 levels of relevancy: 0 not relevant, 1 partially relevant, and 2 fully relevant. Next, please. So this is what the COVID question answering user interface looks like with the user feedback form. The user can type their questions at the top. There's the list of the articles, and then the sentence is highlighted in context and then can provide us feedback. And of course, the system was evaluated, and they are in the process of publishing the results. Next, please, and that will be my last slide. So this effort was part of the Natural Virtual Biomolecular Laboratory, which was an effort that the Office of Science, which is the DOE Office of Science, was asked to the labs to get together and produce this collaboration, and 9 labs actually were able to produce collaboration, and they've done incredible amount of work, which is not I did not speak to. We only spoke to our part in it, but they also produced a lot of characterizations of the protein on the synchrotron. There was many simulations done at extremely high speed, and I refer you to the URL at the bottom for NVBL, which will give you a good example of all the work that was produced at DOE during the pandemic last year. And this is my last slide. So if you have any questions, please either put them in the chat or ask directly. Thank you.
Thank you. That was interesting. Very really good talk. Sorry about the glitches. I didn't realize it would show the screen. I'll hold, as I said, we'll hold off questions at this stage because there are a few more talks to get through. So next up, Can we ask Graham Winter to speak?
Give me just a second. I
Hi,
shall
Graham.
pop of sharing my screen. So I'm assuming that you can see my screen now.
Yes. Yeah.
Yes, you
So
can. Good.
I will just.
Thank you very much. Right. Okay. So yeah, I was asked to come along and talk a little bit about some work we did actually just over a year ago now of sharing some of the very early data we took at Diamond from the COVID-19 main protease studies. So essentially, we had a driver of this, which was Frank von Delft, who's a principal B-Mine scientist at Diamond, wanted to make a fairly large amount of data that we had collected in quite a big hurry at Diamond public with the necessary metadata, with links to, so this is the raw data, with the links to the published structures that had been developed fairly automatically as well. And basically said, here's where the data are, here's a spreadsheet of the information you need, go. And so what the challenge was basically to figure out a way of packaging up the data to make it useful to people, linking it all up correctly and making it public. So, I mean, we've had a number of workshops about, fair access to data and so forth. Actually, in MX, for those of you who don't know, fair data goes back kind of, 50 years now, because the Protein Data Bank was started 50 years ago. And the idea there is we publish the atomic coordinates of structures that you publish, so that users or other scientists and other people can actually download the atomic coordinates and reuse them in a variety of ways. And this comes back to some of the issues raised in the earlier topics, because a lot of the people using these are not people who are sort of synchrotron users. They are structural biologists or biochemists or whatever, and so they need to make sure they've got a good idea of what the data are like. So the PDB, or Protein Data Bank, are actually is 3 different sites who sort of look after the data, and they do a lot of validation on it to make sure that the protein structures are relatively truthful, and we know what sort of quality they are. There's a lot of data and metadata included in all of this, so you can see who collected the data when, on what facility, and so forth. And each PDB deposition is identified by a unique 4-letter name. So, 5REZ or whatever. So, this has been going on for a long time. Sharing the raw data is a topic that comes up in conversation roughly every 18 months, I'd say, but has not really been resolved because the raw data are many orders of magnitude larger than these reduced experimental data. So, at the time, a year ago, the choices I made were basically, why don't we use Zenodo? Because it's actually something I'd already been using for some other activities. It's an EU-funded facility. It's built on top of the Large Hadron Collider Tier 1 data storage, I believe. And it's very, very easy to use, very user-friendly, and available for all of the kind of, a large number of different science areas. So essentially what I did is wrote, what those of us of a certain age would recognize as a mail merge, which takes the sort of spreadsheet that Frank gave me and a load of sort of pre-existing data or metadata that applies to all of them. And then basically goes away, finds the actual experimental data, makes a zip file, and then used some sort of scripts I wrote using Zenodo's API to basically push the data and metadata and create all of the kind of entries, the likes of which you see on the right here. And so the idea was basically we just pushed up all of this data and made it public, and basically any user can come along, get the raw data and reprocess and reevaluate the structures that we've made public. So obviously the key thing with this is not just making the link one way, but you want the link to be bidirectional. So the PDB have the ability to add a DOI to any PDB deposition that includes the raw data link. So what I actually did was ended up sending a list to the PDB curators saying, so basically for each PDB ID, here is a DOI that goes with it to allow you to make the link easily findable by our end users. And I could already put a PDB link into the Zenodo DOI. So that way the link is bidirectional. So it's very easy to go from one to the other. And actually, if you just Google PDB ID and then Zenodo, you'll probably find the right raw data anyway. So to the end users, it's actually very easily findable. All of the kind of experimental metadata is already in the raw data because we use Nexus format. We use standard format. Actually, these data are in CBF format that includes all the experimental metadata. So there's actually nothing more that the average structural biologist would need to go and collect, download some of this data and reprocess it with just information they have online. So from a user perspective, what we were aiming for is actually easy usability rather than, for example, computational efficiency. So although zip files may be slightly unwieldy, they are very familiar to everyone, rather than using a sort of a higher compression format that is less portable would actually probably be a bit more of a barrier. So all the data are in a standard format and they can be processed with standard software in the field. There's no need for a site file or anything. So basically, you can just take the data that's public and reproduce the results that are available. So in MX, the other thing I would say is because the PDB has been around for 50 years, actually the PDB ID is already well understood as being a kind of a unique identifier. So actually people are very familiar with just getting that PDB structure, linking it back to, and so forth. So from the kind of fair and open perspective, that actually made it very straightforward for our end users. So with the depositions, to try and make it easier to find, what we actually did is linked it with the PDB ID, but also keywords like SARS-CoV-2 and so forth to make it easy to find. One of the huge advantages of pushing stuff to Zenodo is it actually, there's no need for any kind of user registration. All you've got is a simple web download. You click on a link and it comes down. So actually, again, we were trying to reduce the barriers to entry as far as possible. And so this was actually a very easy way of getting the job done. I would also say that at the time, uploading big zip files like this, so they're typically 2.5 gigabytes, was not that straightforward with the open Zenodo APIs. And they actually worked quite closely with me to allow me to use some of their unpublished APIs to make the data available. And that basically is all I have to present. It was a big effort a couple of years back, or a year ago, sorry. It was a lot of enthusiasm in the field at the time. I wouldn't like to say how much long-term benefit there was to it, because actually it all went very quiet afterwards. But as a principle, it worked, and it showed that actually making data public, provided that you've captured the relevant metadata at the time of the experiment, is incredibly straightforward. So that's actually all I'd have to say on that topic.
Thank you very much for that really excellent talk, and and thanks for being brief as well. I think it's a little bit of time. Okay, in that case, I think we should move on to our next talk, which is by Jean-Luc Antoni from ESRF.
Yeah. Hello, everyone. Can you hear me? Yes. Okay. So just set up my share here, and then here, and then.
Yeah,
voila.
that looks good.
You're seeing my slides?
That looks good. Yes.
Does the virus look moving? Yes. Okay. So I will briefly talk about the coronavirus structural task force, which has been let's say, community effort to provide some feedback on the early released coordinates from all the proteins from the SARS-CoV-2. I speak in behalf of all the collaboration, and the people will be presented in the end. So SARS-CoV-2 is more than a virus. We have the virion, which is what we usually get represented. This virion is formed by lipids and by 4 structural proteins, but it also contains RNA, which is then decoded by the host cell into a polypeptide, which is cleaved into 16 non-structural proteins. More and more proteins and virions can create it. It's packed inside the cell, to reproduce new virions. And above all, the RNA also codes for 8 unknown structure protein, what were usually called accessory proteins. Up to yesterday, the number have been updated. We have 1,421 structures from all the proteins in the virus, which have been solved by X-ray crystallography, NMR, cryo-electron microscopy, and 2 by neutron crystallography. I've seen John in the audience, and he was involved in the study. So 1,017 by X-ray crystallography. For all the 16 protein, there is at least a part which has been solved, except for the RNA polymerase. 31, mostly the small envelope proteins and the other membrane proteins of the virus by NMR, and 371 by cryo-electron microscopy, the most important being the spike, the RNA polymerase, and the complex of the non-structural proteins 7 and 8. And here is the plot of how these structures were released over time over the last year. So the first SARS-CoV-2 structure was solved in February of 2020. There were then about 100 on average, around 100 released per month, with a big spike around here corresponding to the drug screening from Diamond on this one. I think is the one on the non-structural protein number 3. And somewhere here in the middle, there is also all the data released that Graham presented just before. Proteins include drugs target, like the main protease, which was, again, the example that Graham showed in the previous talk, which is one of the simplest way to block the virus reproduction in the host cell, and so is the target of drugs, or at least a possible target for drugs. Or the spike, for example, which has been in the talks in the last few weeks for all the, let's say, appearance of different variants of the virus. And the spike is the main target of vaccine studies. How do we solve structures? So there's the biological preparation, data are measured, we create a model, and we try then to answer biological question with the model, which is the best explanation possible for the data we obtain. And the model is then used by what we call downstream users, method, for example, computational studies on and this kind of experiments. One interesting thing actually is that more than 90% of PDB downloads apparently come from people who are not structural biologists to start with. The problem, the structure, the fact is that they can contain problems, but they're usually not considered carefully when used in other modeling studies, for example. And this could lead, but we have this example, but it's not important. In crystallography, what we do, for example, we measure the electron density in an indirect way. We measure the diffraction, we calculate the amplitudes, we perform a Fourier transform, and we obtain the electron density, inside which then we trace a model for the protein. So we get the amplitudes from the X-ray data, and from the model, we get the phases to perform the Fourier transform, which leads to a model bias, obviously. We put our model in our initial phases, and our density will then look more like our model. We have ways to calculate this because we use what is called an R factor that tells us how similar my model, how well my model explains the data. Typical R values for small molecule crystallography is about 5%. For macromolecules is around 20%. And fact is that it's impossible to fit a perfect structure inside the protein because there is so many variables inside, and as we've seen, the data inside against which we are fitting are changing along with our modeling process. So completely automation of this is not yet possible, and expertise in many areas is important. Because again, models are mostly built manually. Here comes the task force. We are a group of method developers in structural biology. And what we started from the beginning is to evaluate all the published structure from the both SARS-CoV and SARS-CoV-2. We started with all the SARS-CoV structure from the first SARS epidemic because we thought that will be a good target for first computational studies. And we also checked out selected human interaction partners proteins. And we perform reprocessing of the data when possible, thanks to efforts like the one presented before, and remodel structures when we found that bad problems were possible inside the published coordinates, and we did scientific dissemination, public outreach. So what we did first, we have a pipeline that harvests the new PDB entry by comparing the sequence of each protein, and then we automatically spread the proteins into different folders in our repository, one per protein and one per virus. And if the raw data are available, we also gather the link and put it together with our data. Then we run an automated evaluation that contains many standard crystallographic software provided by members of the task force. So for example, mole probity. We download again the PDB redo results, which is an automated improvement of the model. We check for eventual presence of artifacts in the data like ice rings. And we also run for cryo-EM structures and automated CCP-EM validation pipeline. And then we have a series of experts that have run re-refinement, so have taken the coordinates which were released and tried to improve the models against these. And everything is obviously released in our GitHub, which I will link then in the shared notes. And then on Twitter, we wrote Proteopedia pages, we have a home-page, etc., etc. Modeling problems. There can be many different. For example, a zinc missing in the structure 6W9C, wrong bond chirality in one glycosidic bond, which can be easily fit, or something a bit more problematic. This is an example from our collaborator Tristan Kroll, and it shows how in the RNA polymerase, the first structure, double clicked. If you look for a second, you can see how the structure can be modeled, and now you will see that this model actually fits way better if we slide the sequence back. And there we found that there was a big segment which was lagging like 15 residues back in the sequence. And since this protein had been used for computational studies, most of these studies had then to be redone at a big cost of computational effort, of course. So this is our webpage, and where we publish articles where we explain the role of each of the proteins inside the virus and some of the drugs that have been proposed by the time. We have a lot of partners that I will not list because the time is running short, probably. We produced the first, what we call the first accurate 3D model printing that is publicly available for outreach for the virus. Because normally when you see a model, it looks like a sphere with around spikes which are either too big or too short or too few. And you can follow us on the internet. And this is all the people involved. So starting from Andrea Thorn, who was the, let's say, she was basically the founder of the whole effort. And as you've seen, we gather a quite large group of people, mainly young scientists or early training students. With some exception, we have Dale Torrond and we have the Jefferson Lab from the US. And well, so on a summary, it's important to understand the virus and its life cycle. And to understand it, we need to understand its molecular biology, because this helps in the design of therapeutics. We helped in evaluating this structure both automatically and manually, because structural biology obviously remained difficult, and errors are very easy to do because the models and the data are not perfect. And there's an important question that, at least for us, let's say early career scientists, is important to ask at some moment is, how's the impact of these kind of projects into the evaluation of our career efforts? Because this is 1 year project that is very difficult to put in advance when applying somewhere, for example. And with this, I would like to acknowledge first Thomas, who did all the nice pictures that you've seen in the presentation. Our collaborators, the Iwasa Lab at University of Utah, also do visualization, and the funding by the DFG. And thank you all for your attention.
Thank you, Gianluca. Good to see the model of the coronavirus. This is mine. Okay. In the thank you for that. So, as I said, please hold off questions, because we've got a couple more talks to do. So, please welcome our next speaker, who is Darren Sherrill from the Advanced Photon Source, who thank you for getting up very early, and also Lynn very early for coming to speak to us today.
Hello. Okay. I'm standing in for Nick Schwartz. He's basically head of IT for APS at Argonne. But I'm going to give a more sort of example of my experience with the COVID-19 and sort of, metadata and how we do one of our experiments as an example of boots on the ground type of science. Okay, can you see my screen? I can't see any of you. If someone says no.
We can see it in not in presenter mode. We can see the presenter view. That's good. That's it. That's presentation view.
Excellent. Okay, so I got a pretty good inside look at Argonne and to some extent Diamond ALS and ORNL during this experience. We had, I want to give historical credit first because the work that I do stands on the shoulders of a lot of other people, and it started sort of 6 years ago, my type of experiment that I performed. So I don't want to just give credit to these people first, which is the Dwayne Miller group out of Canada and Germany, the Huff's group out of England, and then Ilma Schuttling's group out of Germany. So when COVID hit Argonne, we went from 3,500 people at the facility to 35 of us. And some of those were security guards and cleaners. And so most of these were protein crystallographers working out of CSGID. And they were basically, there's a facility right next to APS called the Advanced Protein Characterization Facility. And the crystallographers in there were basically purifying protein as rapidly as possible. Our first experiment was actually a remote experiment on January 28th of a SARS-like virus from SARS-CoV-1. But then during sort of March and April, we were one of the people producing these early structures. And it was at my beamline. And my beam, it was interesting because it was very important work, but I had to do exactly nothing, which was I could make sure the beamline didn't break. And to make sure your beamline doesn't break, you usually just don't touch it. So I spent many months not touching my beamline while these structures were being developed using normal, regular single crystal crystallography. And they're still producing them now. And you can visit the CSGID website and you can see, what proteins have been done and published and where we are. I'm a methods developer person. So I do a type of crystallography called serial crystallography, and I'm going to talk about that and how we do that experiment. And this is related to FAIR by sort of like the best practices that we use. And I didn't even know that I was using FAIR because the IT people were using FAIR, and I was just a recipient of all this thoughtful stuff from previous work. So serial crystallography, instead of shooting one crystal, we shoot thousands of crystals, just a little bit. And that way we reduce the overall radiation on any one crystal. So the radiation damage to the structure is reduced. There's also an introductory method for when you want to do time-resolved crystallography, which basically allows you to do sort of structural dynamics and get these sort of molecular movies. Well, that's the promise anyway. We developed something called the Alex holder. You don't really need to know much about it. It just holds thousands of crystals. This is actually my co-worker Alex, and I named it after him because he developed it. So I work at 19ID, and this is just how it looks. It's just a nylon mesh, and in the nylon mesh you have a slurry of crystals, and then you're just shooting thousands of positions in this nylon mesh. It's pretty crude, and one of the good things about it is sort of mindlessly repetitive. And when you do these experiments, you have, here's the ADRP. It's just the diffraction pattern that Gianluca was talking about earlier. So the thing about serial crystallography, it's mindlessly repetitive. So, and it's also on known structures. So this crystal's already been solved. So you have your model already, but what you want to do is take all these thousands of diffraction patterns and you have this known model. And so what I developed was basically a flowchart for the IT people so that when the data comes out of the back of the detector, it gets taken up into the cloud, goes off to the supercomputer, and then they take my knowledge that is automatically ingested in the metadata. And I'd like to speak to what Abigail was talking about earlier about what is the point of the metadata. And for me, as a scientist, I feel like I do two things. I do experiments and I publish my data. And so for me, the metadata is useful in two ways. One, it's so that I can remove myself from the actual analysis. If I can train a computer to do what I do, then I don't have to do it. And so that's a useful thing for metadata to have. And also publishing. When I go back and I have to publish my data, I want all my metadata to help me with my publication for my tables and to keep track of what I actually did. And for me, those are the two most important things for metadata, is basically try and take myself out of the analysis loop and also come back and be able to find all my data and find all the everything that I did with that experiment. And so I can report that accurately, because someone told me that a publication should really be a way of teaching another scientist how to do what you just did. And I think metadata is important for that. This is a simplified version of the flowchart where we collect the data at the top. We take these still images, and the most important thing we need to know is whether we want to continue to take more data or move on to the next. And basically, we're trying to train the computer to do this instead of us. To do this, I've used the Globus Automate Cloud Service. Globus is out of Chicago, I think, and they're related. There's a big group of them here. And I realized that my Beamline didn't have the compute resources that I needed. And I went to a talk and I saw these, and it turns out that Globus doesn't just do endpoints for data transfers. They also have a couple of other tools like FuncX and Automate, which you can actually make your computer either locally or remotely do a type of analysis that you ask of it. And it's a really, really useful tool that I found. They helped me a lot, and we're working closely with them. My ultimate goal for this is to actually be able to take data, serial crystallography data anywhere in the world, and then transfer it to any compute resource in the world, and for it to go through the flowchart and do the analysis for me. So I can basically take my experiments to exactly the right beamline or synchrotron or XPEL that I need, and then compute my structures as quickly as possible without actually any intervention from me at all. This is just a screenshot from Theta. This is the supercomputer, and when you're using it, it can tell you, which nodes you're using. It's fun to look at when you're doing your experiment. It's more complicated. Another thing that FuncX and Automate does, when they ingest all your information, they actually publish it into a portal, and then they can work on your data and give you feedback information. So here we can see maps of, 2 of the nylon meshes, and basically what you're looking at is a red dot is there's 1 crystal in that position, an orange dot is there's 2 crystals in that position, and then a yellow dot is that there were 3 crystals in that position. And this just tells you how many, basically how many crystals were where on your nylon mesh. And then the data is published as well, so you can search it for it like you're buying something on eBay. And this sort of searchable, where FAIR comes in with this published and searchable information with metadata included, is I benefited hugely from this, just so I can peruse my data and find out information and look at best practices for what I want to do for my next experiment. And that all comes from Globus basically enforcing best practices on their work. We had really good success. We discovered we found the methylated form of the NSP10, NSP16 protein complex, which is the methylation is basically the way it hides from the host by hiding, I guess. This is probably the end slide. No, we'll just go back one. We'll leave it at success, and I'll finish there. Ours is just an example of how we did a COVID experiment, and I hope that was quick enough.
Thank you very much. It's in the chat. There are some questions in the chat, but I think we should. I think we can answer them, but maybe we should move on with the presentations just to finish off, because we are beginning to run out of time. So thank you very much, Darren. And now our final presentation is Katharina Lauer from Resear, who's going to be sort of moving away again from the facilities directly, talking about the COVID-19 data portal.
Yeah, hello everyone. I hope you can hear me well.
Yes, you can.
Yeah, I realized that I'm maybe a bit the odd one out here. So for all of you who don't know me, my name is Kathy Lauer. I work for Elixir, and I joined Elixir actually 3 years ago. And I had previously trained as a virologist with a specialization in vaccine design, and then decided that there wasn't a lot of excitement in that field. And turned into more like data-driven applications, bioinformatics. And then, of course, last year, I was all of a sudden able to combine both, and that's why I'm talking to you today. So let me just share my screen. Right, so you should see my slides now, hopefully, in presentation mode.
Yep, they're there now.
Fabulous. Good. So yeah, I'll tell you a little bit about the European COVID data platform. And yeah, so just to give you a little overview of how we got started. So like everyone else, we were all hit a bit by surprise when SARS-CoV-2 really took off last year. And we kind of got together with the European Bioinformatics Institute and with the European Commission and said, well, we need to do something. That was at the beginning of last year. And then around April time, we actually managed to gather all resources that would help to set up that COVID-19 data platform. So we just had the anniversary last week. And yeah, so what is the COVID-19 data platform? It is a platform that unites various different databases, for example, around protein structures, around genomic sequences, and brings them all together in a one-stop shop. And over the course of the last year, we've added more and more and more of those different services and resources to that one platform. And as you can see, we've seen a steady growth of data coming into the services that are affiliated with the platform. So the platform consists of 3 components. This is the COVID-19 data portal, which is run by the European Bioinformatics Institute. And I'll show you the actual data portal later. We look at that. It consists of something called the SARS-CoV-2 data hubs, which are local instances for genomic virus data in each country, in each organization. Sometimes, you can't really necessarily make them directly available in an open database. So this is the reason why those are generated separately. And then the third component of the data platform is the federated EGA, which is a database for sensitive human data to be deposited, and it can then be shared in a federated way. Right, so this kind of explains a little bit the data flow into the COVID-19 data portal. So on the left, the data producers, this can be, for example, universities, this can be hospitals, medical centers, so coming from all sorts of different backgrounds. And they produce the data, and then that data needs to flow into a database. So the different data types can be quite diverse. For example, expression data sets, protein sequences, genome sequences, etc. And of course, you can't establish all those databases ad hoc. So many of the databases that are combined in the COVID-19 data portal have been around for many, many years and even decades, and have been established, for example, by the European Bioinformatics Institute. So, yeah. these data producers deposit their data into those specific databases, which are then exposed in the COVID-19 data portal. So this is the open research data side, and you can see those pink arrows going into the portal. Then we've got also the opportunity to deposit access controlled data into the COVID-19 data portal. These are either, as I mentioned, viral data sequences that come in through those SARS-CoV-2 data hubs, or this can be sensitive human or clinical data coming in through the federated EGA, and there is a way of access controlling that data. And within the COVID-19 data portal, this one-stop shop, users can access and find the data. So I've just introduced you to the COVID-19 data platform run by the EBI, but there are several national instances. Sometimes a country needs to run a local instance, either because their data can't leave the country, or there is information specific to researchers in their country, or, for example, there's a language barrier. Not many countries prefer to have their local instance in their local language. So the EBI helped to set up those local instances of the COVID-19 data portals, and we'll look at those later in a bit more detail. So I want to shift gear a bit and talk a bit more about open and fair data in the context of the COVID-19 data portal. So, I don't have to tell people here why it is important, but considering we've been working with the European Commission very closely on establishing the COVID-19 data portal, I just wanted to highlight that there's a general sharing policy to share data as open as possible and as closed as necessary, and share data in line with the FAIR principles. Particularly then, with a focus on COVID-19 research, there was a strong emphasis of making data accessible without delay, shareable and reusable whenever possible. And we've really tried to incorporate that as much as possible in the COVID-19 data portal, for example, through specific search functions, make data accessible, for example, through APIs, and make it, for example, reusable, which is a problem with many other databases, through clear licensing, etc. Yeah, I already said that before. I mean, I probably don't have to tell you the benefits of sharing, particularly during a pandemic. Not benefit, for example, when doing research, getting ahead of your competitors. This is not really the aim in a pandemic. In a pandemic, we need to share and collaborate to really benefit everyone globally, and hopefully manage to move beyond that pandemic soon. So collaborative working is really encouraged through the data platform. And I mean, I talked a lot about the data inflow and data usage, but many of our collaborators have worked tirelessly last year to develop, analysis tools and really help users to actively be able to reuse and collaborate using the data deposited in the data platform. In that regard, I want to point out an open letter on COVID-19 data sharing that we've published a couple of months back on the COVID-19 data platform. Please have a look and see if this is something you can support as well. I think it's really important that we who believe in fair and open data sharing talk about this as widely and openly as possible. Okay, so thank you very much for listening to me. And I thought I'm just gonna quickly show you actually what the data portal looks like. So when you arrive at the data portal, you see the landing page. And as I mentioned before, just last week we had our anniversary, so you can read everything about the history of the portal here. You can see that it is divided up in different data type groups. So you can, for example, look at protein structures or viral sequences. And yeah, so here you can see the list then of sequences, different data types. You can, for example, have a look at the statistics if you're interested in seeing who is depositing data. Yeah, where does it come from, which countries, etc. You can, I mentioned that before, you can have a look at all the local instances of these national data portals. And I've raised that before. Sometimes it is important that a country has their national instance of a portal. For example, let's take the Japanese one. A lot of researchers there might not be actually able to, you know, follow all the information as clearly as it should be if it's not in their local language. So it's really important we have those local instances. And of course, they make different things available in their national portal. You can see related resources. And this is actually something this is also pointing into what the future vision for this data portal is. We're working very closely with the European Bioinformatics Institute to bring in various different data types that are maybe not represented in the portal at the moment. So at the moment, you see a lot of biomolecular data, but in the future, we want to bring in social science data, epidemiology data, etc. And you can see, for example, there is a start here with the French epidemiology portal. So I invite you all to have a look and see if there is anything that is useful to you. I guess particularly proteins imaging is something that's of interest to this community. And if you need any help in submitting data, go to submit your data and you will see the list of affiliated resources and how you can get support and help. Thanks. That's all from me.
Thank you, and thank you for hurrying at the end. We're about to crash the end of our time, unfortunately, and we haven't managed it. So thanks for that really good, fantastic presentation, and thanks to all our speakers for their really fantastic presentation. We haven't had much chance to have discussion, but I thought it was good to get all our speakers to have the opportunity to speak. We've had a lot of discussion and chat in the questions answered in the chat. I think there was a really follow-up question, which is from Frank, which was, you know, our users are probably very unaware of those data sharing recommendations, and are there ways we can raise our awareness of our community? So I think that's something we would want to follow up with after this. So I think, you know, we're now. Oh, Frank's coming in. Oh, we've not been thrown out completely. So maybe Frank, you can say a word or two.
Actually, I thought this question actually is. Goes to Katharina as well, because the guys here working at the Beamline, they have not the faintest clue about the COVID-19 data portal or what they can actually expect now and deposit there. So it's exactly the same question, how to actually raise awareness of these kind of things, which are extremely useful.
Well, I have to say, generally, my experience is that we as researchers are really bad at communicating, unfortunately, and we don't want to listen to the communications people when they tell us how to communicate those things so everyone understands what we're doing. So I don't know how we can get beyond that, but I guess if everyone makes a little effort and, you know, tells their colleagues about the kind of resources that are available there, or just takes 5 minutes to have a look and see how your own research activities, your services can feed in. And the people at the EBI are very helpful. They have a support help desk. You can leave feedback. You know, if there is anything you want to be reflected in the portal, just reach out and tell them. They sometimes don't know by themselves. But other than that, I guess we just all have to become better in communicating. and listening to our communications people, I guess.
I wonder whether there's anything we can do either in this group or in the projects that we're involved with that might be able to help that activity. So I think that's something we would want to discuss offline. And if there's any people here who would want to be engaged in that discussion, please contact the group, or contact me, or contact the other members of the group, and I think we will look to follow that up. So I'm now where we are over time. So I would like to thank you for joining us this afternoon. I think there's been really lively discussions in the chat box. I hope, sorry we haven't managed to maybe answer all your questions. Please add notes to the Google Doc and your name if you are willing to share it. I'll put that up again in the chat. Thanks to the speakers, particularly those who joined very early from the US. And thanks for Rob and your comment on the community doing FAIR. I think that our community has made quite a lot of progress towards FAIR, and I think we've seen some really good examples of good practice. I think there's more. Still think there's more that we can do to do it better. So with that, I would like to propose we close the session. And thank you very much, and have a great rest of your day.